Right, then hello everybody. So my name is Ben. I'm currently a PhD student in my second year at the University of Vienna, Institute of Mathematics, and the Vienna Graduate School of Population Genetics. Today I'll talk, as was introduced, about the external branch length distribution, mainly with a simulation approach. As part of that, I've also considered the so-called singleton density score. There are a bit of background noises. Oh, thank you. I will also consider the singleton density score, which is basically a direct application of the external branch length distribution in a selection test. The basis of the external branch length distribution is, of course, the coalescent, the so-called n-coalescent. As probably all of you know, the basic n-coalescent describes the past neutral evolution of a present-day sample of n gene copies. And this asymptotic result of the n-coalescent developed by Kingman is valid for various breeding structures or various probability models. And hence it can be derived for various forward models, be it the White-Fisher or the Moran model, as a limiting process when the population size grows infinite and the sample size n stays fixed. Therefore, it's a very universal result and of high interest to study. The output of such basic coalescent is obviously a gene genealogy, which is just a random discrete tree topology and a sample of independent random continuous coalescence times. With that, we can therefore describe the entire ancestral relationships in our n sample. On top of such a genealogy, which I'm already showing here, of course, you can drop mutations. In this presentation, I'll only consider the infinite sites model, and they are therefore independent. And if a genealogy represents a single site or SNP, then clearly just a single mutation can be placed on such a tree structure. We will here consider ancestral states, therefore 0, and derived states 1. And I'm showing here such example coalescence tree with 10 samples on the left-hand side and 5 samples on the right-hand side. Since this will be predominantly about theoretical population genetics and not phylogenetics at its basic sense, we are not necessarily interested in the tree structure, as may be the case in phylogenomics or phylogenetics, but here I'm rather interested in the coalescence times, and namely the branch lengths that give rise to particular variation in our sample. Okay. All right. So to finalize this introduction, of course, for the basic coalescent, we require a population that is much bigger than the sample size, small n. Here, n describes the effective diploid population size and not the haploid population size. As usual, time is measured in 2n generations, so-called coalescence units. And we see tree structures are of different shape when we're varying our sample size n, as can be shown in this depiction. All right, so we have therefore introduced the coalescent process, and we of course know that simulating this process is rather easy and highly efficient. So we can take a present-day sample of n gene copies and just keep coalescing the currently available ancestral lineages randomly using a randomly drawn exponential distributed coalescence time. This process is just iterated from n-1 until 2 from this point onwards, and thereby we create our full genealogy, a rooted bifurcating tree for the basic coalescent. So we now have a simulation approach that is actually very efficient and therefore can be computed quickly. And we also should therefore consider what is possible on top of just the basic coalescent with neutral evolution. And in principle, we can say there are sort of partial expansions that consider several additional population genetics factors. So obviously, drift is considered in the basic coalescent already, which is the randomness in birth or death. Mutation is also considered. And now a new thing is, of course, recombination. So how does the genealogy change across a chromosome when there are recombination rates between neighboring loci. There is one concept, the so-called ancestral recombination graph, where now the word tree was exchanged to a graph, as it turned into a more complicated structure, because now it's not a tree anymore, but you may have circles inside your graph due to recombination. But this is fairly well considered, so recombination is included in the form of this, for instance, ancestral recombination graph. On top of that, there are also various results that consider demography. So if you consider, for instance, population increase, decrease, so changes in N over time, or population structure, like particular island models, etc., there are analytical results available. But for this, already results are mainly partial, and you need to be lucky that you find something that fits your needs. This is so important that it has its own name, namely the structured coalescent. The last point that is remaining and of high interest, obviously, in particular for selection tests, is natural selection. And for this, only for very special cases, we can have analytical formulas that use coalescence theory to make particular predictions about the outcome. Okay. What is now the difference between coalescence considering selection? Well, it means that differences now in reproductive success are correlated with genetic differences, so inherited. If, however, differences are there, but not inherited, the basic coalescent can actually still accommodate that. But we are not concerned with the latter, but with actual inherited differences. All right, so we learned backward simulations are extremely efficient, but they do not cover the full range of scenarios we are interested in. Therefore, I'm interested in finding additional approaches that give the same output to me, but allow me to consider different evolutionary scenarios. So besides backward simulation, for instance, by Hudson's algorithm, we can also just forward simulate an evolutionary simulation and obtain a gene genealogy from that. In the past, this was rather difficult, particularly due to computational inefficiencies. But for this, we now just consider we forward simulate a Wright-Fisher type of simulation, where of course multiple mergers are possible, and we record the emerging tree structure. And afterwards, throw everything away except the tree structure of our extant population, where we may of course subsample the extant population to a particular sample size n and study its properties. So of course, the structure is as in any other coalescence tree. If we just look at a single SNP, at a so-called marginal tree, namely for this particular example, we have 9 external branches because this equates to our sample size that we chose, 7 internal branches because it's n-2 always. And we now can look back until the most recent common ancestor and study the particular branch lengths in this tree, which then tell us about the properties in terms of genetic variation, also upon selection. In particular, I'm considering the infinite sites model of mutations. Therefore, for a single marginal genealogy, I only place a single mutation. Okay, that's for that. So we now learned that therefore I can extract branch lengths, but in particular, I labeled my talk with the word as singletons. So what is a singleton mutation? Well, for a singleton, we need to consider only the external branches. Every coalescent tree has n of those. And the distribution of these branches is of course constrained by the structure of the tree. But for a neutral model, it's quite easy to extract some values for that. So if we call, for instance, tau I, the total length of branches that have I descendants in the sample, so then tau 1 is of course the total length of branches that give rise to the total n descendants, sorry, 1 descendant. So the branches that can give rise to singleton mutations, the only branches namely. Then we can easily find the expected value of its length in coalescent units. Basically, it's 2. And just to compare, we also can easily find the number of singletons, namely looking at the first value of our unfolded side frequency spectrum, xi I, namely just by multiplying theta half times this expected total time of branches that give rise to a single descendant, which is then just theta for nu, which is quite obvious. Singletons are the most prominent polymorphisms, as it's basically easily seen from the shape of the side frequency spectrum. After that come doubletons, etc. So what happens now under selection? Well, if we consider a single SNP with some ancestral allele A1 in a diploid population, then we may let a derived allele A2 occur in a single individual, which then sweeps according to a hard selective sweep model. We can therefore decompose the population into two subgroups, the ones carrying the first and the ones carrying the second allele. And we know that if a subpopulation is declining, namely the ancestral allele, then of course we get an excess of middle frequency SNPs because of this sort of bottlenecking going on. And if it is growing, then this is the case at the derived allele. We of course get a classical shape from a hard selective sweep with an excess low frequency SNPs in our sample. Right. So what about recombination now? So we already considered the effect of selection. Well, considering again our diploid Wright-Fisher model, Hudson already provided an algorithm to consider recombination back until the most recent common ancestor at every marginal genealogy, which is just a subtree of the full ARG, which goes back to the grand MRCA. So we therefore now consider marginal trees along a chromosome, and therefore trees neighboring each other, between which we therefore have some recombination possibility, are different by particular tree operations. So they are correlated. Closer trees are more similar to each other and require less tree operations to make them equal. All right. So now coming to the external branch length distribution analytically, there exist results if there is no selection going on. I'm showing, for instance, one for the total branch length that gives rise to single descendants. And I'm just showing here in this curve different approximations by different authors over in terms of coalescent time and their respective density. All right. So there are approximations available for the neutral model, but not for selection. All right. Clearly, if it is drawn. Okay. So now we learned that we can simulate directional selection using forward individual-based simulations and extract the genealogy. And this may, of course, include recombination, as I've introduced. So therefore, the approach I use to study that is that I conduct individual-based simulations with SLIM, and I use the so-called methodology of tree sequence recording. This is very efficient and therefore overcomes the barrier of computational inefficiency and allows us now to construct the genealogy while simulating forward in time extremely fast. On top of that, it also provides a very efficient data structure as output, namely a sequence of correlated trees along all sides of our simulated chromosome. So how I simulate is as follows. I start, here we look at the middle, where this line, where this arrow is. There I basically start a forward simulation, for instance, of Wright-Fisher type, with, for instance, here, a hard selective sweep, namely a single chromosome carries a mutation at a single locus. After finishing this simulation, I get an extant sample, which is at the bottom, where present is written. And now having this extant population, I only trace back towards the initial generation, namely the initial generation only for those individuals that are actually leaving descendants in a final generation. And for those individuals, I can now just on top namely backward in time, attach a coalescent process. So I may, for instance, consider a neutral coalescent by assuming that, of course, the selected site did not exist before. And therefore, I'm basically conducting a very efficient simulation, leaving out all unnecessities. On top of that, I then further process my data by using particular libraries in Python that allow me to handle tree structures. And this tree structure is equivalent for a coalescence simulator, for instance, MS Prime, and therefore allows easy interchange and comparison of statistics. Here, I'm now showing you the singleton density score from the paper of Field et al. So this is a score that allows a test to test back for very recent selection, namely in the last 2NE over n generations back in time. And the key insight is that the tip branch length or external branch length distributions are much shorter for the derived allele, for the selected allele, here shown in light blue, than for the ancestral allele, which is not selected. And therefore, we can easily extract a statistical test that considers the distance away from a particular SNP to the left and right, where the next singleton is found. And this distance has to be greater, on average, for the selected site, as the rate at which singletons are created, which is now dependent on the tau1, is smaller. All right, I will skip the closer background on the singleton density score, but it's basically just on top of that, building a likelihood model, extracting a test statistic.
You have about 3 minutes left.
Yes, and thank you. Building a neutral model for neutral simulations and then comparing it against an arbitrary selection model to find selected sites. Now I'm showing you just a few preliminary results that consider this approach I'm using. I'm using a diploid population, for instance, here of one non-recombining locus. And I simulate here a sample of 10 chromosomes, basically with a coalescence simulator. Then I can easily study the distribution of external branch length branch lengths, shown here on the left, for the simulation. And here I'm displaying real generations, not coalescent units, so to say. Right next to it, I have a theoretical result extracted from Calibre that is rather involved, so I don't mention it here, which is basically exactly overlaid to the simulation results. And this provides the starting point for my investigations. Basically, I can now do selection scenarios. So here I present one for the same population size, 51 loci, and in the middle, basically at the point 25.5, I place a selected site with selection coefficient 0.05. And I now study how the external branch length distributions is modified. And you see here the cumulative density function, or step function in this case, where you compare the ancestral allele in red versus the derived allele in green. And you see that really, for the derived allele, the expected branch length is much shorter, which is as expected. And now I can go on by introducing linkage of different levels and studying this distribution again. And not only that, but also, as you see here from top to bottom, by considering sites neighboring the selected site of 25.5 to study the decay with ongoing recombination. And you see, if we just look at this figure, here the effect is still pronounced, as you see in the differences in the cumulative distribution. There's a high probability that it's a higher probability that the value is smaller for the derived allele than for the ancestral allele. This effect is nearly disappearing and now nearly disappeared when we go just 2 positions away. Okay, that's for that. And now I would like to conclude. So in my work until now, I have built an approach that allows me to study this external branch length distribution. And in particular, I'm interested in doing that for polygenic adaptation. So I want to study branch spectra when there is not only one selected site, but several. On top of that, a singleton density score so far does not consider recombination. So I'm interested in if I can also extract signals from recombination, because this also, of course, carries information about selection. And because since it only considers singletons, I only also want to include higher n tons, like double tons, into the test. And with that, I would like to close, and I thank for your attention.